next up, as we talk about natural history studies and where all your data has gone to, I'm really excited that Elli Brimble is going to be leading this session. Some of you met her last night. As I mentioned, she was Amara's genetic counselor and has been with the foundation day zero before we had a foundation, and is running all of our clinical work, connecting to our clinicians, et cetera. So she's going to be going through the panel of our data, and here she is.
I think so. Hello. All right, well, it is such an honor to be here with all of you today. Thank you for allowing me to tag along into this community and this wonderful journey. It's been the honor of a lifetime and career defining, so thank you very much. Disclosures, I work at Invitae. So I wanted to spend a little bit of time before we get into the data breaking down what is a clinical trial and why do we feel that the work that we're doing with the natural history data is important. So we can think of a clinical trial in two components. We have an intervention, which might be a drug, like what Allison was talking about, or a therapy. But then we also have a measure of effect, so something that we can use to determine whether or not that drug or that intervention is doing anything meaningful. And you might hear that measure of effect referred to a few times today as an endpoint or a clinical endpoint. So a clinical endpoint is an outcome that can be measured. It might be something like a finding on a physical exam, a laboratory result, a developmental milestone. If you look at any of the gene therapies or ASO therapy trials that have been done in other disorders, these will look familiar to you. But importantly, it should represent a desired outcome and measures how a person feels, functions, or survives. And it should be something meaningful to the person or a surrogate for something meaningful. So it's not very helpful to demonstrate change in a feature that people don't really care about or do not feel to be problematic. That said, it can be pretty hard to identify these relevant clinical endpoints in rare disorders. So as everyone in this room knows today, meeting each other's children, rare disorders are incredibly variable. There is often a limited understanding of the disease course, so you may notice if you go to Google FOXG1 syndrome or read any papers about FOXG1 syndrome, they tend to be giving you information at a point in time, but we don't have very much information that understands how does this condition look across the lifespan and how do things change and evolve over time. There can often be limited understanding of the biological pathways. And traditionally, there is limited engagement with the patient community. And so, this challenge of wanting to make sure we're changing the things that the community cares about can be really difficult to do if we're not engaging with that community. So this is where the natural history study comes in, which, per the FDA, is a pre-planned observational study intended to track the course of disease. Its purpose is to identify the demographic, genetic, environmental, and other variables that correlate with the disease development and outcomes. So natural history studies are really important in rare disease, and some have stated that they are a pillar of epidemiologic research in rare conditions. So we can use natural history studies to inform endpoint selection, guide clinical trial planning and design to make sure that we're setting ourselves up for success. They can be used as an external control arm in a clinical trial. So in many cases, if you want to have a very rigorous trial, you would compare individuals who receive a placebo, which is not the drug, to individuals who did receive the drug. But we know in rare disease that might not be ethical because it's not fair for some people to receive a drug and others not to. And so our hope is that we can actually use natural history data or historical data to serve as that control so that every child can receive the drug in a trial. We can also use it to identify research priorities. So these are studies that are focused on patients and outcomes, and so we can use them to understand what are the most critical things for us to focus our funding and our research efforts on. And lastly, we also hope that these studies can contribute to the development of care guidelines. So a common theme that you'll hear about today is many individuals who receive a diagnosis have a care team made up of wonderful physicians who have unfortunately never heard of FOXG1 syndrome before. And so we want to be able to use our clinical data to help make sure that everyone who receives a diagnosis of FOXG1 has access to recommendations for the expert management of that disorder. And then lastly, we may also be able to use this to develop centers of excellence where individuals with specific expertise in FOXG1 syndrome and multi-system expertise in FOXG1 syndrome come together so that children can have expert and coordinated management. And so, when we talk a little bit about today, what we're hoping to do is really parallel process. So Thursday was fantastic. It was so inspiring and just really validating to see all of the wonderful work that has gone into developing therapeutics. But we really want to parallel this with making sure we're identifying clinical endpoints in parallel for this concept of clinical trial readiness. So, to be most efficient at the time that we identify candidate therapies, we also want to know how to measure them so that we can move into the clinical trial space faster and we don't need to wait to figure out what the endpoints are going to be moving forward. So this is why we're really focused on this now, even before we have a candidate therapy in sort of these pre-IND enabling studies. So I hope I've at least managed to convince you that natural history studies are really important, but one challenge is that these are really hard to do in rare disorders, so they're very expensive to run. It's challenging to have families who have children with significant physical disabilities to come onto a clinical site every few months for assessments, especially over many years. You'll note that Allison mentioned that the Angelman Syndrome Natural History Study was 15 years. That's a long time to sign up for annual visits. The travel can be incredibly burdensome, especially if your child has devices that are required and you're flying into sites. The prolonged duration, they take too long. And then there's also a question around data accessibility. So one thing we've observed in the traditional model is that as participants of those studies, it can be difficult to actually get insights into what you are learning from that data. And then when it comes time to share that data with others in efforts to accelerate drug development across multiple companies, that can also get a little bit fishy too with like data transfer agreements and things like that. So these are really important. That is undisputed, but it is challenging to do in rare. And unfortunately, at the time that we realized that we needed to do a natural history study, we didn't have enough money. So that's when we were like, we need to come up with another approach here because we need to start doing this now when we don't have time to try and raise, $3 million to get this done. So we ended up applying for funding for a grant from the Chan Zuckerberg Initiative, which we received. And the goal there was to use a patient-centric real-world data platform to generate natural history study data in FOXG1 syndrome with a very specific goal of characterizing and selecting endpoints. So there are a lot of really interesting things that we can learn about the physical and clinical manifestations of FOXG1 syndrome, but we wanted to make sure that we were incredibly focused on achieving our goal of clinical trial readiness. And so we're very fortunate to receive that funding as part of the RAROS1 program through CZI. And I'm going to talk to you a little bit about the outcomes of the first couple cohorts of the study. So we are planning to pair retrospective medical record collection. So if you have joined the Citizen platform and indicated where you have received care, this is why we are actually collecting all of your medical records. We have years and years of data spread over an average of about 5 different institutions. So we are capturing the full spectrum of your care or your child's care. And we are pairing that with prospective survey and validated measure assessments over a period of 3 to 6 months, or rather they're repeated every 3 to 6 months, so that we can pull the objective data from your medical records, but also make sure that we are capturing the subjective experience longitudinally. And so you'll notice, if you haven't participated yet, that some of these surveys that you will receive talk about things like developmental outcomes or capabilities. These are kind of 4 different ways of looking at that. Behaviors, and then also sleep, which is intended, again, to understand some of the features that we have heard from families are important. And then we're actually about to hit a really exciting milestone, which, in the pandemic, time has lost all meaning, so I don't actually think I realized it until we were putting these slides together. But we are going to be reaching our final study time point for the first cohort of 50 individuals this month. So super exciting, and we're hoping to be able to share that data more broadly. And then just really quickly to talk about the Citizen platform, which is what we are using to generate this real-world data, is that this is a platform that allows users to collect and store their medical records, and then Citizen is developing technology to translate those medical records into a research-ready data set, meaning that when we share that information with people, they don't need to do any cleaning or organization of the data to make it make sense. It's just ready to go. And then in terms of what we capture, we really focus on longitudinal phenotypes. So this includes things like diagnoses, symptoms, signs that are reported, and then also the ways that those things are managed. So that could be medications, surgeries, injections, and many other variables as well. So really trying to represent these conditions over the course of an individual's lifespan and not just at a specific snapshot. So diving into the data, we have 2 cohorts of 50 individuals now who are enrolled, so a total of 100 patients, which is incredible. A huge thank you to this community for rallying around this effort. This is one of the most successful recruitments that we've seen on Citizen across all of the rare conditions on the platform, so huge kudos to this community. For our analysis, we get kind of like some annoying questions from people sometimes. So one of the ways that we try to mitigate that is to focus on individuals who only have a change in FOXG1 syndrome, FOXG1 the gene. So we do have some individuals on the platform who we have data for, but for this analysis, we have excluded them if their deletion or duplication contains genes outside of just FOXG1. So these are larger copy number variants. And then we're just showing down the breakdown, showing the breakdown of the cohort here. So when we look at the different types of FOXG1 variants, these are the numbers that are observed in our cohort of 90. So you'll notice that about half have frame shift variants, followed by nonsense and missense as the second and third most frequent. And then we do have a few individuals with in-frame deletions and one who has a gene deletion of just the FOXG1 gene. And the average age of our cohort is about 7.5 years of age, and we have medical record data for an average of 7 years of data. So we feel like we're doing a really good job at capturing the full lifespan. And then you'll note too that we actually have pretty good distribution across the United States, so that's wonderful as well. And then just to note that for genotype-phenotype correlations that we'll talk about today, so that's mapping clinical outcomes to a particular variant type, like frame shift, nonsense, or missense, we are focusing on the 3 where there are more than 5 individuals, and there were no changes or no differences in the age makeup of those 3 cohorts. So this is just showing the distribution and frequency of genetic variants in our cohorts. And so this is the FOXG1 amino acid sequence. So this is essentially the protein of FOXG1 that is made and functions in the body to perform the job that FOXG1 is supposed to be doing. And what we have on the very top of this are all of the frame shift, nonsense, and in-frame deletion variants. And so you'll notice that we have a few specific letters within the FOXG1 protein sequence where we have a number of individuals who have changes at those particular regions. And so you'll see here, for example, the most common variant is a frame shift variant at the 154th letter. And then we also have the missense variants tracking along the bottom of the protein. And one thing that I think you'll all probably notice is that we have a really strong restriction of missense variants to a very particular region of the FOXG1 protein. And we know that this region is really important for one of the key jobs that FOXG1 does, which is binding DNA to regulate the expression, so whether other genes are turned on or off in the body. So this has been a really interesting phenomenon that we've known about in FOXG1 syndrome, but is replicated in our cohort as well. So when we looked at the full cohort in order to understand what were some of the common symptoms that we were observing, these are the features that were present in more than half of the population, and this is showing when they were first diagnosed. So if we look along this horizontal axis at the bottom, this is the age in months, and then each individual dot represents one patient, and it is showing the time at diagnosis. And then they're ordered by the time that they appear on average. So we can see that things like microcephaly and developmental delay appear relatively early in life, so before 1 year of age. And then if we go down the list, we can see that there are other conditions that tend to be diagnosed later in life. So for example, movement disorder is one of the more common conditions that we see, but it tends not to be diagnosed until the child is a little bit older. So this can give us some sense of when we might expect certain symptoms to emerge, but also what are some common symptoms to make sure that we're screening for and looking for. We can also look at the relative frequency by the specific type of FOXG1 variant. So in this case, I've made it hard for you. I've switched to the axes. So now the different symptoms like microcephaly, strabismus, which is having crossed eyes, hypotonia, which is low muscle tone, these are all indicated down along the bottom axis. And then on this vertical axis, this is showing the frequency of individuals with those particular symptoms. And so the first bar are individuals with frame shift variants. The second bar are individuals with nonsense variants. The third bar are individuals with missense variants. And then the fourth bar is the entire cohort of 90 individuals. And so one thing that we observe is that for many of the symptoms that we are observing in FOXG1, it does appear that individuals with missense variants have lower frequencies of those particular symptoms, whereas individuals with frame shift or nonsense variants may have higher frequencies of those conditions. So it doesn't mean that the risk is there or it is not there. They just appear to be slightly different frequencies depending on the type of variant. But as we know, every child is unique and we're proven wrong pretty frequently. So as I mentioned, lots of cool things that we could do with the data, and we definitely want to get to that at some point, but really our priority is making sure that we understand what are meaningful things that we can measure potentially in a clinical trial. And the way that we prioritize these is to look at other clinical trials. So for other rare disorders that have had successful therapies, or clinical trials rather, what did they measure, and can we potentially just copy that to be more efficient? But then we also, the surveys that everyone completes in the registry, we use those to inform what is important to families, and we wanted to make sure we represented those broadly. So we looked at epilepsy, movement disorder, developmental outcomes, sleep, and then healthcare utilization, which is a measure of how often is someone interacting with the healthcare system. That obviously has a burden on families, but from a more cynical point of view, insurance companies care about this because it is expensive to interact with the healthcare system. So starting with epilepsy, which it includes seizures as a feature, we found that epilepsy was present in about 70% of the cohort. And this really ranged from about 50% in individuals with missense variants up to about 3/4 of individuals with frame shift variants. And on average, we saw that there was childhood onset of epilepsy across all genotypes. So this is a similar graph to the first one that I showed, where we have age along the x-axis in months. And then these are the different genotype breakdowns where each little dot represents a single patient. And so what we can see is that sort of the bulk of diagnoses of epilepsy are actually occurring at 12 months of age or after. And we also noticed an interesting trend, and just keep in mind that these are small numbers, so we want to continue to recruit and revisit this over time. But one thing we did notice of interest is that it looked like individuals with nonsense variants were less likely to have seizures in the first year of life compared to the other genotypes, where we see many dots within this first gap between 0 and 12 months of age. So that was something that we observed in the data related to epilepsy. So we wanted, again, to focus on measurement, and so we were looking for ways to not only say, do you have epilepsy or do you not have epilepsy, but are there ways that we can measure the relative severity? Are there things that we could potentially count over time? And so we chose to look at two different things. One was the number of seizures that were reported, and then the other was the number of concurrent medications that a patient was using to treat seizures. So we know that seizure medications are not the most fun medications to take. And so even if you have relatively well-controlled epilepsy, if you're on like 3 or 4 seizure medications, that's still impacting your quality of life. And so we wanted to make sure that we were capturing that as well. So the first row of data here is looking at the relative frequency of seizures. So this is counting seizures over time. The first graph is showing the maximum seizure frequency score observed either across the whole cohort in this bottom row or the 3 different genotypes here along the top. And what we saw was that sort of the average maximum scores were between 4 and 5, which is consistent with having at least daily seizures. So this represents a pretty high seizure burden on average for each individual with epilepsy. And then what we were trying to illustrate here with this heat map is how does that change over time? So again, as we're moving horizontally, this is showing increasing age in months, or rather in years of age. And then each of these individual rows represents one of the genotypes. And then if we look at the colors, they start to show us a picture. So these darker purple colors have lower seizure frequency scores, meaning that you are having fewer seizures, and then the brighter yellow colors are showing higher frequencies, so more seizures, more daily seizures, or having multiple seizures per day. And so a couple things I wanted to point out. Again, we see the same pattern if we're looking at the 0 to 1 year of age range, where individuals with nonsense variants actually have significantly fewer seizures in the first year of life compared to individuals with other types of variants in FOXG1. So that kind of just validates the information that I showed in the previous slide. But I think the other thing that I wanted to call out is there doesn't appear to be a noticeable pattern that is occurring over time. It looks like on average people continue to have seizures, and so it continues to be an active issue that is managed for these patients. And then on the bottom now, we're looking at the number of seizure medications used at the same period of time. So if, again, we look at the average maximum, we're looking at between 2 and 3 seizure medications on average used at the same time per patient. And then again, looking at the heat map, this is showing how the number of concurrent seizure medications used changes over time or changes as people age. And I think one thing that is interesting here is we do see concordance, right? So when we see that the colors are getting brighter, we do see kind of brighter colors matching on the number of seizure medications, which is good. It suggests that people who are having more seizures are being treated for those seizures. But what I also wanted to point out is even when there are periods of relatively good seizure control, like we can see here for the older individuals with missense variants, those people are still being managed for their seizures. And so that's great. It suggests the seizure medications are working, but the patient is still needing to take seizure medication. So that's helpful for us to know. So moving on to a movement disorder phenotype, we wanted to look at these longitudinally, and so the way that we did this was by looking at all of the neurology exams that patients had in our medical record data. And so when we looked just at the number of exams per participant, on average, we were seeing that each person had around 2 exams per year, but there was certainly some variation. And then when we looked at the average number of exams that had an abnormal movement finding, about 50% of exams reported that. In order to look at how this changes over time, we looked at the percent of exams within a particular age period, so 0, 1, 2, 3, 4, etc., years of age. What percent of those exams had an abnormal movement finding, and then we wanted to break that down by genotype as well. So the legend here shows you which line belongs to which cohort. And I think undisputably, we are generally seeing an upward trend, right? So as children are getting older, we're seeing a higher frequency of movement findings on their physical exams. And one thing to point out is that by 3 years of age, half of exams will have an abnormal movement finding. And then we also note that individuals with nonsense and missense variants by about 8 or 9 years of age all have abnormal movement findings on their exam. So this suggests that this is a relatively prevalent finding in FOXG1 syndrome. And then when we look at the most common types of movement findings that are reported on exam, these include chorea, dystonia, hyperkinesis, stereotypies, involuntary movements, and orofacial dyskinesias. So if you're ever looking at medical records, you might notice some of these words coming out on exam findings. So we actually have a few different ways of looking at developmental outcomes, but what I wanted to share with the group today was the results of the Vineland survey, which participants of our natural history study are completing. And so the Vineland measures different aspects of development or activities of daily living, and I'm sure many of you have done this either through school or through clinic or through our study. The information that I'm showing today is largely developmental quotients. And so this is a calculation that you can do from the Vineland data where you're comparing the age equivalent. So how does your child perform on the test? What is the equivalent age? And then we can compare that back to their chronologic age. So for example, if your child is 3 years of age, and then on the Vineland, their age equivalence comes back at around 2 years of age, then we can actually compare those numbers to compute a developmental quotient. So in this first set of graphs, we're looking at 2 domains of the Vineland related to language or communication. So on the first graph, we're looking at receptive communication, and then on the second, we're looking at expressive communication. So how do you receive and how do you share communication? And what we found is that in general, most people have delays in these domains. So their age equivalencies are lower than their chronologic age. But we also saw some differences between the different types of variants. So we observed that for expressive language in particular, individuals with missense variants had significantly higher developmental quotients than individuals with frameshift or nonsense variants. So that suggests that their age equivalence on the test for missense variants was a little bit closer to their chronologic age. But again, there was some variability here. We also looked at different measures of motor function. So we have gross motor on the left and fine motor on the right. And what we observed again is that individuals with nonsense variants tended to have lower developmental quotients compared to the other variant types for gross motor. And then in fine motor, missense variants tended to be associated with higher developmental quotients than individuals with nonsense variants. So kind of take-home message here is that across language and motor, we did observe that there were delays across all individuals with FOXG1 syndrome, and we did observe some slight differences depending on the specific type of FOXG1 variant. One thing we wanted to investigate was whether there was a correlation between development and the presence of epilepsy. So epilepsy is a very common endpoint that's used in clinical trials because seizures are countable, and so they're kind of easy and the FDA recognizes it. But when we've talked to families, seizures are not always the first thing that families ask about, right? In many cases, families actually talk to us about developmental outcomes like communication. And so we wanted to look at whether people with or without epilepsy had higher or lower developmental quotients on the Vineland across the different domains that we tested. And what we did find that this was true for gross motor and fine motor in particular. So I'm showing 2 different measures from the Vineland here for gross motor and for fine motor function. And we see that the first one and the third one are the developmental quotients. So those are those scores that we've been talking about. And then the second and the fourth are growth scale values, which are just a different type of calculation that you can do on the Vineland that is better suited to tracking change over time. And these little asterisks here above the lines comparing mean that there is a significant change. So we are observing a pattern in the data that is unlikely to be happening by random chance alone. And what we're showing is that individuals with epilepsy across gross motor and fine motor domains of the Vineland tend to have lower scores than individuals who do not have epilepsy. It is possible that some of this may be driven by genotype as well, so it's something we want to continue to look into as we have more Vinelands completed over time. But we thought that this was interesting, that perhaps even if we are measuring epilepsy, it might have implications for other phenotypes that are important for FOXG1 to measure as well. So when we talked about there being like a surrogate endpoint, perhaps epilepsy could also be a surrogate for developmental outcomes too. We also looked at sleep. So we used a questionnaire that measures sleep challenges. And when we score these questionnaires, in general, scores above 40 are indicative of sleep problems. And so we observed that on average, most people had scores consistent with there being problems with sleep. And then when we looked individually, more than 75% of respondents had a score indicating that there were challenges with sleep. And in support of that, when we looked to the medical record data, we saw that more than 50% of participants in the natural history study also were taking a medication for sleep. So they were being managed medically either with melatonin or CBD or clonazepam to help manage their sleep. And then lastly, we also looked at interactions with the healthcare system. So this first is showing the number of procedures, so either diagnostic procedures like an EEG or a brain MRI, and also therapeutic procedures like a surgery or an injection, for example. And on average, we saw that each individual had about 13 different diagnostic procedures done, and there was also an average of about 2 therapeutic procedures done per participant. And then we also looked at hospital admissions, which we know can be frequent, particularly in the first couple of years of life. And so we saw that on average, each individual had about 4 hospital admissions, and this translated to about 15 and a half days admitted in total. So this is kind of aggregate data, which we think is really helpful for conveying, what does FOXG1 syndrome look like on average? But we were worried that it doesn't do a great job at actually conveying what does it mean to live with FOXG1 syndrome. And one of the things we're really excited about with the citizen data that we're generating is we can actually show this. And so we're always looking for better ways to do this. And this is something that I put together that I hope will resonate with many of you here today. But this is actually looking at one participant that I really just chose at random, but I think ends up doing a really beautiful job at illustrating FOXG1 syndrome. And so each of these dots represent different events or things that we were tracking in the citizen data. And then again, along the x-axis is their age in months. So the first dark purple line are diagnoses, followed by EEGs, brain imaging, cardiac screening, surgeries, and then hospital admissions. And so we can see that we start to collect data for this participant around 2 months of age, and they are a male who presents with microcephaly, so a smaller head and brain size, global developmental delay, and abnormal movements. And there is concern at this time that those abnormal movements may actually be epilepsy, so he is admitted to the hospital for a workup. So we can kind of map that to the events here where we're seeing these new diagnoses emerging. They have an EEG, imaging, they have some cardiac screening done, and then they're also admitted to the hospital at this time. He ends up not being diagnosed with epilepsy then and goes home, but at 7 months of age, he does go on to have seizures that are diagnosed as epilepsy. He starts on a medication to manage those seizures. And during that time, he also has genetic testing performed, which diagnoses him with FOXG1 syndrome. During this time, he has ongoing feeding challenges and failure to thrive, meaning that he's not gaining weight in the way that his physicians would hope he is. And so he has 2 surgeries performed. A G-tube is placed and a Nissen fundoplication at 8 months of age, which is represented by this green dot. What I think really stands out to me is we see that during this period of time between about 1 and 2 years of age, he has many, many hospital admissions. And this is largely to manage his epilepsy. So he's having breakthrough seizures even on his medications. And in total, he will spend 76 days of that year in the hospital, which I can only imagine the burden that is to the family. We see here that he has a VNS implanted around 30 months of age, and this is after trying 11 different seizure medications and the ketogenic diet. Fortunately, the VNS is associated with a significant reduction in seizures. And then as he gets older, he goes on to have a number of other symptoms diagnosed that we know become hallmark features of FOXG1 syndrome. So he is diagnosed with sleep problems, strabismus, chorea, stereotypies, intellectual disabilities, reflux, and scoliosis with time. And then here I just wanted to show the overlay of the seizure frequency, so really showing how many different data points we are able to capture related to seizure frequency, and to overlay those with all of the different medications and diet and surgeries that he was trying. And so we see that this VNS implantation around 30 months of age, we see a dramatic reduction in seizures after that procedure, so much so that we also see that he is able to be weaned from some medications, but continues to be on 3 different therapies for seizures, including the VNS at the time of his enrollment in the Citizen platform. So even though we have really good seizure control, he's still heavily managed with polypharmacy. So what we hope we're showing is that real-world data can support longitudinal characterization of FOXG1 syndrome, and I'm hoping that we're also really able to demonstrate that there is a significant unmet need in this population. It is not acceptable that we do not have better therapeutics for FOXG1 syndrome, and that is what we are working to change. And we also hope that with the work that we're doing today, we can continue to highlight candidate endpoints for clinical trials and to characterize those, and also demonstrate feasibility of being able to track those in a clinical trial. And so with that, a huge, huge thank you to this community and all of the participants. I hope that we're continue to able to demonstrate the incredible value of your participation. Thank you to our incredible advisory board, a few of who are here today, for their wisdom and expertise and mentorship as we work through this. And then, of course, to all of our funders. Thank you.